Like many scientists, neuroscientists try to devise experiments to understand how the brain works. And in my case, our interest is in trying to understand how the brain develops at early stages of embryonic development. It's almost magical because the brain self-assembles. It puts itself together. There's no one in there doing it for us. It self-assembles at very early stages. And it's been wonderful to understand that there's actually an orderly process going on here. The first stage of this involves proliferation of neurons to generate massive numbers of cells. This is followed by a period of migration when the cells move to their final locations to take up their final dwelling places. And then there's an extended period of differentiation when the neurons acquire the functions that they will play in the adult brain. So we now have some understanding of how this process occurs in general, and we are doing experiments to try to figure out how these processes are regulated during development. It's difficult to work on the human brain for several reasons. First of all, it develops in the uterus when the embryo is developing, and as a consequence, it's not very easily accessible at the early stages of development. And then on top of all that, it develops rather slowly over a period of months. So we've chosen to work on what's called popularly in science a model system, the frog nervous system. And this is quite attractive because the fertilization of the eggs occurs externally, typically in pond water. Here, of course, we do it artificially in the lab. And then the embryos are accessible throughout the entire period of development as a consequence. In addition, the maturation of the embryos is quite rapid, and the nervous system effectively is fully blown, fully developed within 24 hours after fertilization of the egg. So it's quite a rapid process. In biology generally, and neuroscience specifically, different model systems are best suited for the solution of particular problems. And so a whole variety of different species are in active use today, from fruit flies to nematode worms, zebrafish, frogs, which are our model, mice, rats, and so on. All of these have proven useful in different ways. There are different advantages and also disadvantages to each of these different model systems. Because biological systems are related to one another, the expectation is, and indeed the proof has been, that discoveries made in a model system are often generally applicable to the human condition. And so this is a benefit to working with a model system. It's implicit in the studies of development that if we understand how development normally occurs in the nervous system, then we can hope to be able to prevent disorders of development of the nervous system, a very important objective. And this is one of the goals of the National Institutes of Health that provides funding for our research and for the research of many other investigators. The development of particular tools have been crucial to scientific advances in all fields, and certainly neuroscience is no exception. The development of electron microscopy that became available in a major way in the 1960s really gave us enormous insights into the structure of neurons. In the 70s, techniques were developed for recording the electrical activity of single neurons. Again, very powerful, giving us an understanding of how individual nerve cells behave. With the development of fluorescent probes that one could use to tag particular proteins, look at the expression of particular genes, there was a real revolution in our ability to address scientific problems, understand how nerve cells develop and how they function in the mature nervous system. And most recently, in the 90s, the development of brain scan technologies have been terribly useful, understanding in the intact animal and in the intact human being how our brains are acting while we are doing particular attacks and perceiving particular objects in our environment. These tools and others that are still to be developed will allow us to address questions, probably some of which we haven't even learned to ask at this point. Scientific problems become ripe for solution, just the way fruit becomes ripe for picking. And the development and practical use of new tools is an important aspect of this ripeness and readiness for solution of scientific problems. I have to say that it is tremendous fun and can be very exciting to be discovering something new that no one had known before. This is a deeply satisfying and very rewarding process.